Bienvenidos al ciclo de conferencias de investigación Universidades por la Ciencia, organizado por Fundación UNAM, el Colegio Nacional y el Consorcio Universidades por la Ciencia. Les agradecemos el interés y que nos acompañen. Y el día de hoy tenemos una conferencia muy interesante a cargo de la doctora Sara TISHKOFF de la Universidad de Pensilvania. La conferencia inaugura un ciclo sobre paleogenómica, genética, y es uno de los temas de investigación que se caracteriza por una muy rápida evolución, un muy rápido desarrollo. Los cambios y los avances que se dan es cada vez en tiempos más cortos. Clave de la vida y cómo evolucionan las especies y evoluciona también los humanos. El ciclo de conferencias es organizado y coordinado por las doctoras Mashal Sohail y Araxi Urrutia, quienes han construido realmente un programa extremadamente interesante. Welcome, thanks very much, and welcome to the University for Science Conference Series. Dr. Sara TISHKOFF, a professor in the Department of Genetics and Biology in the University of Pennsylvania. Thanks very much, Dr. TISHKOFF, for accepting this invitation. Well, we are very grateful. And Dr. TISHKOFF is a specialist in genetics, genomics, and the evolution of the humans. And this is a research field that is very rapidly changing and advancing. And we are extremely pleased. And today, Dr. TISHKOFF will present the results of the studies in the African continent. We are extremely excited. And this will inaugurate the series of conferences, this cycle. The conference cycle has been very successful. We have been having presentations on very different disciplines for several years. And this cycle was part of the International Year of Basic Sciences by the United Nations and the UNESCO. And is also a part of the priorities of the UNAM Foundation. We have also the company of our president of the UNAM Foundation, Dionisio Meade, the executive director of the UNAM Foundation, Araceli Rodriguez, and Araxi Urrutia is also the president of the Consortium for Universities of Science. Gracias nuevamente, muy bienvenidos, y vamos a pasar a la presentación de la doctora TISHKOFF y de la parte de la serie. Muy bienvenidos todos. Y pasamos a nuestro presidente de Fundación UNAM, Dionisio Meade.
Muchas gracias, Jaime. Muchas gracias a todos quienes participan en este evento. Agradecemos de manera especial a Marshall Sohail y a la doctora TISHKOFF que nos acompañan y que pues harán de esta serie una presentación seguramente formidable. Estamos muy agradecidos, gracias a Araxi por convocarlos, y pues queremos mencionar que esta serie organizada por Fundación UNAM y por el Colegio Nacional está dentro del marco del Consorcio de Universidades por la Ciencia. Y este consorcio, de manera informal, se constituyó pues como una respuesta a las dificultades de comunicación directa que se generaban a través en el momento de la pandemia, y fue una respuesta para poder difundir la ciencia, la investigación y el desarrollo. La verdad ha sido muy exitoso, ya tenemos casi 5 años haciendo estas presentaciones, y eso nos ha permitido formar parte del comité asesor que estableció Naciones Unidas para impulsar las ciencias básicas. Y primero fue 1 año y ahora es 1 década, y se ha considerado por la UNESCO, por Naciones Unidas, que este es un medio eficaz para promover y divulgar la ciencia. Así que nos da mucho gusto participar en este esfuerzo, y nos da gusto también porque se abre la posibilidad de que muchos de nuestros jóvenes, los propios mexicanos y quienes forman parte del consorcio puedan participar en este evento, y quienes estén por definir su vocación, su interés profesional, puedan hacerlo a través de estas presentaciones. Así que pues por todas estas razones nos motiva de manera particular la realización de este evento. Agradecemos a los organizadores y agradecemos a la doctora Mashal y a la doctora TISHKOFF que estén acompañándonos y presentándonos pues esta investigación. Se trata siempre de investigaciones en curso y pues esperamos siempre, y seguramente será el caso, de que las preguntas de quienes participan sean muy motivadoras del interés que está despertando una presentación como esta. Muchas gracias a todos, muy buenas tardes, y pues con la mejor disposición de escuchar las presentaciones que el día de hoy nos van a hacer pues estos académicos formidables que hoy nos acompañan de la Universidad de Pensilvania y que refuerzan pues esta visión que el consorcio tiene de que sumemos a muchos en favor de la educación superior. Gracias a todos y muy buenas tardes.
Muchas gracias. Nos estamos vistiendo de gala el día de hoy. Estamos iniciando y arrancando un ciclo de 10 conferencias sobre genética humana y tenemos el gran placer de que la doctora Mashal Sohail colaboró con el consorcio, con Fundación y con el colegio para poner esta lista de magníficos ponentes. Ella nos va a dar, de hecho, la última presentación. Ella cierra esta serie que concluye en 10 semanas. Entonces cerramos con broche de oro y también abrimos de la mejor manera con una excelente presentación. On behalf of all of the institutions that form the University for Science Consortium, we appreciate your presence and we are very, very pleased to present to you this series of 10 public talks. We are starting with a great presentation. In a minute, Dr. Mashal Sohail will introduce our speaker for today, and we'll have great presentations all along the next 10 weeks. Some of them in English, some of them in Spanish, but I hope all of them with subtitles. La doctora Mashal Sohail realizó sus estudios de licenciatura en el MIT y después sus estudios de doctorado y maestría cursó de manera concurrente en Harvard. Entonces, bueno, creo que eso básicamente dice todo acerca de su talento y la calidad y la estatura que tiene como científica, y es una fortuna para la Universidad Nacional Autónoma de México contarla hoy entre su lista de académicos. Ella trabaja y lidera su grupo de investigación en el Centro de Ciencias Genómicas. Muchas gracias, Mashal. Dr. Mashal Sohail, who put together this stellar list of speakers that we'll have the pleasure to hear over the next 10 weeks, did her undergrad degree at MIT and then her postgraduate studies at Harvard University, and today she leads her research group at the Center for Genomic Sciences at the National University in Mexico. Many thanks.
Muchas gracias, Araxi, Jaime y Meade. Es un gusto estar aquí organizando este ciclo. Buenas tardes a todos y todas. Hoy tenemos el placer de tener la doctora Sarah TISHKOFF con nosotros. So today we have our first speaker of the cycle, Dr. Sarah TISHKOFF. She is the David and Lynn Selfin University Professor in Genetics and Biology at the University of Pennsylvania, holding appointments in the Perelman School of Medicine and the School of Arts and Sciences. She's also the director of the Penn Center for Global Genomics and Health Equity in the Perelman School of Medicine. Dr. TISHKOFF studies genomic and phenotypic variation in ethnically diverse Africans, and her research combines fieldwork, laboratory research, and computational methods to examine African population history, the genetic basis of anthropometric, cardiovascular, metabolic, and immune-related traits, and how humans have adapted to diverse environments and diet. She has played an active role as an advocate for the inclusion of global populations in human genetics and genomics research. And she's a member of the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. So it's a huge pleasure and honor to have her here today. And without further ado, she will be presenting to us the first talk in the series on African evolutionary genomics, reconstructing human evolution, and the genetic basis of complex adaptive traits. Please, Sarah.
Well, thank you all for the wonderful introductions and the warm welcome. I am truly honored to be here today. Let me go ahead and share my screen and we will go ahead and get started. Okay, and let me put this in presenter mode, one moment. And there we go. So today, I'm going to tell you about our research studying genomic variation in Africa and what that can tell us about learning more about human evolution and how we've adapted to different environments throughout human evolutionary history in Africa. And I thought I would start by talking about what I think are some of the key challenges in human genomics research today. So the first is that we need to do a much better job characterizing both genomic and phenotypic variation, meaning trait variation, in globally diverse populations. We want to try to understand the evolutionary processes that generate and maintain that variation. We want to understand how gene-protein, and gene environment interactions contribute both to normal variation and disease risk. So the focus of my research for the past 25 years has been on Africans, and there are a number of reasons for that, but one of which is the fact that all modern humans, anatomically modern humans, originated in Africa. These red dots represent the locations of fossils of anatomically modern humans, the oldest of which is dated to around 300,000 years ago. This was found in Morocco. Following this origin in Africa, somewhere, let's say, between about 60,000 to 80,000 years ago, relatively small numbers of people, could have been in the hundreds, but no more than in the thousands, left Africa and migrated across the rest of the world. We now know that when modern humans left Africa, they ran into, and it turns out that they interbred with, archaic populations such as Neanderthals, who were in this geographic distribution, and another group called the Denisovans, who are really interesting because they've actually been identified only from ancient DNA that was found from a pinky bone. And so we know that they have interbred such that somewhere between around 1% to around 6% of the genomes of non-Africans derive from those archaic populations. So how much do we differ at the level of the genome? If we were to compare genomes between identical twins, in theory, there shouldn't be any differences. If we compare the genomes of random, unrelated humans, we should differ at about 1 out of 1,000 nucleotides. If we compare human to chimp, it's about 1 out of 100. Human versus mouse is about 1 out of 30. And if we compare humans to broccoli, it's about 2 out of 3. So given that the entire genome consists of about 3 billion DNA bases or nucleotides, that means roughly 3 million differences between, say, a pair of genomes. Now, actually, this is a slide I borrowed from Lynn Jordy many years ago, before they were doing whole genome sequencing. What's remarkable is that this prediction has held up very well. So this is supposed to be somewhat humorous, representing genomic diversity across ethnically diverse, beautiful people, but even amongst all of us, very, very similar when you look at the entire genome. So less than 0.1% divergence. Now, there's also considerable what we call structural variation. So those are sort of bigger things like big insertions or deletions or duplications or inversions in the genome. And some people estimate they could account for as much as, say, 1 million base pairs of differences between any pair of genomes. We also have known for many, many years that there's more genetic variation within relative to between populations. And all of this makes sense in terms of this relatively recent African origin. We all share this common ancestry not that long ago. And however, that doesn't mean that differences don't exist, because they do. And this was actually taken from the 1,000 Genome Sequencing Project. And this was an effort led by NIH. They actually ended up sequencing more than 1,000. I think it was probably closer to 2,000 from geographically diverse populations. So what we're seeing here is that variants that are shared, they did whole genome sequencing. If they're shared across all the continents, they're shown in this dark gray. If they're shared amongst populations, I believe this is with, let's see, I'm just trying to get this across, all kind of shared across continents. I'm still trying to distinguish those, but basically gray means shared. And then private to a continent would be, for example, yellow here. And then private to a population would be orange. And we could see that there's not that much that's private to a population, but there are some that are private to continents. And again, that could be due just to this history of migration, random mutation that occurs, random fluctuations in frequencies of variants and natural selection to different environments. The other thing that motivates my own research is trying to understand risk factors for health disparities. So, for example, hypertension is more common in individuals of African ancestry in the US. Type 2 diabetes is more common in people of Native American ancestry and African ancestry compared to other ethnic groups. Now, there's no doubt that there are major social demographic factors and things like access to, nutrition, access to healthcare, and things like that are playing a major role in these health disparities. But we think that there may also be some underlying genetic risk factors as well. And one of the biggest challenges is, how do we distinguish the gene by environment interactions that together contribute to these health disparities? As we showed in this paper several years ago, and this still holds true today, in fact, I think it's gotten even worse today, if we look at the catalog of genome-wide association studies, so these are studies where people look at genetic variation across the genome, they look for an association with traits of interest or with disease risk, let's say heart disease, for example. There's a database of all of these, but about 80% of the individuals included in those studies are of European ancestry. About 10% are of Asian ancestry, around 2% African ancestry, and I would say the majority of those are African Americans from the US. And then 1% Hispanic or Latin American, less than 1% everybody else, and that's a real problem for a number of reasons. It's going to impede our ability to fully understand genetic risk factors for human disease, and it could actually exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it could be mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies. And that's because, let's say that there's a patient, someone suspects that there's a genetic disorder. So they sequence the genome and they're trying to find what is the mutation that's causing this disorder. And one of the ways they do this, there are publicly available databases like ClinVar. And one of the criteria for whether or not something is pathogenic or causes disease is how common it is, because the idea is if it's very common, it's unlikely to be really bad for you. But maybe it's not common in people of European ancestry, but it is common in other ethnic groups. So that could actually change the assessment. And then lastly, people nowadays are using estimates of genetic risk from genome-wide association studies. So for example, let's say someone does a study of genetic risk for heart disease, and they find all the variants and they add up, which variants put you more at risk and which ones are protective. And then they go to a patient, let's say, and you look at their genetic variation and you add up these scores to look at their total risk for heart disease, for example. And what has been shown is that when these genetic risk estimates are done in European studies, which is predominantly the case because you need really big sample sizes to have power. And then you try to assess risk in someone who's not European, it does really, really poorly. So that's some of the reasons we need to include diverse populations. And as I said, I am particularly interested in studying Africa. I want to give a little bit of background about African variation. So there are over 2,000 languages spoken in Africa, and these have been classified into 4 major language families based on similarities of those languages. So in blue, this is the distribution of populations that speak Afroasiatic languages. So that would include Semitic and Cushitic languages, for example. In red is the distribution of populations that speak Nilo-Saharan languages. Many of them also happen to practice a pastoralist lifestyle, so they have cows and practice dairying. You might have heard of the Maasai, for example, in East Africa. So they tend to be in Central and East Africa predominantly. There have been some migrations to the West in the past. The largest language family shown in orange here is called Niger-Congo, and it originated in Western Africa. The largest subfamily of those languages are called the Bantu languages. They're thought to have originated around the border of Cameroon and Nigeria. Now, these Niger-Congo populations had developed, say, around 5,000 to 6,000 years ago, iron tool technologies and something called slash and burn agriculture. So with the iron tools, they can go into the forest, they can cut down the trees, they can grow their crops. If they grow crops, they can sustain very large population sizes, and they were very, very successful at that. So over the past 4,000 years or so, there was this massive migration from that region in Central Africa to Eastern Africa and Southern Africa, and to the West into Southern Africa. And that's called the Bantu migration. And after they migrated, they intermarried, admixed together with indigenous groups from those regions so that they really shaped the genomic landscape in Africa today. And then the last language family spoken is classified as Khoisan, and it includes clicks, which I'm not very good at doing, mainly spoken by people called the San, who were, until very recently, have been living a traditional hunting and gathering lifestyle in Southern Africa. And then 2 other groups, the Hadza and the Sandawe. The Hadza in Tanzania, the Hadza still speak, still practice hunting and gathering, and the Sandawe did until just a few hundred years ago. So I wanted to tell you a bit about the field work that we do in Africa, and I want to start by acknowledging all of my collaborators and partners in Africa, because this couldn't be done without them. So everything that I'm going to present to you is done in partnership with my collaborators. So we have predominantly been studying people who are living more traditional lifestyles, living in very remote areas. That means that we have to take all of our supplies with us. We basically are setting up our labs in the bush. And we're very careful to do this research in an ethical manner. So what that means is that we have to get IRB approval, first, University of Pennsylvania. Then we have to go through multiple rounds of ethical review in each country, ultimately getting permission at the national level. But here, for example, this is my collaborator, Dr. Alfred Njamshi from the University of Yaoundé in Cameroon. Our group spent almost a year in advance before we even stepped foot in the field to collect samples, going to the populations. We translate, first we talk in layman's terms, and we translate into the local language. And it's only after we get community consent, and then we ultimately get individual consent, and then we can proceed with the study. We also think it's really important to return the results to participating communities. Again, we do this, we present community level results presented in sort of layman's terms and translated into the local language. And I think it's a form of benefit sharing, and it's a way that people know what you have done with these samples that they have so generously provided. And then training and capacity building is really important. I've had the honor to train a number of grad students and postdocs, and most recent being Tafadzwa, or Taffy she goes by, who's currently in my lab. And with the hope that this knowledge will be brought back to Africa and will lead to research being done locally. So in the field, we measure phenotypic diversity, variable traits, and you can imagine in these really remote areas, it can be challenging. So we're getting very detailed anthropometric traits like height and weight and limb length and grip strength and skin color. We're looking at cardiovascular, lung, and blood traits, blood pressure, oxygen level, hemoglobin, lung capacity, red cell and white cell counts. We look at metabolic function, lactose tolerance test, glucose tolerance test, and we look at infectious disease status when we can. So first I'm going to tell you about a study that we published not long ago in Cell. And this was a study of high coverage whole genome sequencing from 180 Africans from these ethnic groups shown here. There's 15 per population. And what I'm showing you down below, this is called an admixture plot. And we are statistically, probabilistically inferring genetic ancestry, which is represented by the different colors. And each, you can see these are actually made out of lines, and each line represents an individual, and they can have different proportions of ancestry. And then the red arrows are showing the populations that we've included in the study. And what you could see is that this is Western Eurasians, shown here in green. And here are the sub-Saharan Africans. There's a lot of variation in Africa, not just within, but between populations. So we've selected the ones representing these different inferred genetic ancestries. And just looking at the people, you could see there's a lot of cultural diversity, differences in language, differences in diet. So some are hunter-gatherers, some are pastoralists and have cattle and dairying, some are agriculturalists, and so on. So we found over 33 million single nucleotide polymorphisms. That's regions that are variable in the human genome. About 17% were novel at that time. That's just from 180 people. And then of the novel ones, we predicted which ones are likely to be functionally important. And we found a lot, mainly in non-coding regulatory regions, so regions that are regulating the expression of genes, but we found some in coding regions as well. So from these sequences, you can make a phylogenetic tree. And at the end of each of these branches is a genome sequence. We have color-coded based on the language spoken, and then we've listed the name of the ethnic group here to which they belong. And we could see that at the root of this tree, the most basal lineages are those that are in the Click-speaking San populations from Southern Africa. And this is what many other groups have observed as well. We then see the rainforest hunter-gatherer groups from Central Africa, then the Bantu-speaking groups who originated also in Central Africa. Then we see the two Click-speaking groups from Tanzania. And then we have a number of groups that are predominantly from Ethiopia, also the Fulani, who are like a nomadic pastoralist group. And then here we have the non-African populations who have a subset of the variation that we see in Africa, again, consistent with this origin of modern humans in Africa. We then wanted to look at potentially pathogenic variants. And I mentioned this ClinVar database that has these supposedly pathogenic variants. And we found 44 variants that are predicted to be pathogenic, so causing disease potentially. But it turns out that while they're rare outside of Africa, this is from a database called Nomad. And these are mainly non-Africans. Here, AFR means African Americans predominantly. And we could see that most of these variants are pretty rare outside of Africa. They might be at a few percentile, in the African Americans, for example. But in some of the African populations, they're very common. So this is a variant that's associated with the type of hypotension. And in this one population called the Fulani, it's at like 13% frequency. These other three are variants in a gene that have been associated with the type of alopecia. So that's hair loss that's often seen actually in women of African ancestry. And we could see that while it's rare outside of Africa and few percentiles in percentage frequency in African Americans, it can be pretty common in certain African groups. So maybe these aren't pathogenic, or maybe they are and there's some kind of modifying mutations, or maybe they are also adaptive in certain environments. So that's something we want to look into. The next thing we want to know is the level of genomic variation. comparing the genomes to the reference genome. And basically the point here is that the higher up this is, that you see these bars, the more variation there is. And the populations with the most variation are those click-speaking San from Southern Africa, who also have the most basal, the oldest lineages, followed by the rainforest hunter-gatherers, then again, these Bantu-speaking groups. And at the low end are the Amhara from Ethiopia, who actually have a lot of admixture with non-Africans, somewhere around 30 to 40%. And then if we were to put non-Africans here, it'd be down here right around that 3 million that I had mentioned earlier. So a lot of variation in Africa. We then use some computational approaches to make inferences about changes in what's called the effective population size. It's related to the, say, the number of breeding individuals. And again, I just want you to look at the relative numbers. That's the most important thing. So this first approach is most informative for older dates. So say 50,000 years ago, going back in time. And we see that in that time, the populations that had the biggest, the groups of the biggest population size were the click-speaking San, the hunter-gatherers from Southern Africa, and the Central African rainforest hunter-gatherers, the Baka. If we look at more recent events, so this is, going back in the past 10,000 years, we see some really dramatic population crashes in some of the hunter-gatherer groups, the Hads and the Shabu from East Africa. Their census size is only about 1,000. And then some of these other populations have maintained relatively big population sizes. And then together with our collaborator, Yun Song and his group, we use a number of computational approaches to make inferences about the demographic history of Africans. And this is I would say, very simplified version of what we found. And it's a really complex history. So frankly, this could change in the future. It's very challenging to study this data. So what we're calling here a ghost population, it's not really a ghost like you might think of a ghost. It means a population that doesn't exist today, but we see evidence, just like the Neanderthals interbred with modern humans outside of Africa. We think this was a divergent lineage like Neanderthals, almost as divergent as Neanderthals for modern humans, that existed in Africa and interbred with the population that was ancestral to all modern humans. And we think there's been some more recent interbreeding as well in some of the Western African, Central African rainforest hunter-gatherers and Bantu-speaking groups. The other thing to note is that unlike other prior studies, the best fit model that we found showed an ancient common ancestry between rainforest hunter-gatherers and the San populations from Southern Africa, diverging around 80,000 years ago, and that it was a group ancestral to them that gave rise to all other populations. The other thing you can note is this very deep divergence times between groups. And OOA, by the way, refers to out of Africa, the non-Africans. But between African groups, really deep divergence times. And these blue lines represent migration and admixture. And we see evidence of old migration events and then much more recent migration events like the Bantu migration. So the next thing we want to study is how do people adapt to diverse environments? So some of you might have heard, for example, about sickle cell disease. So sickle cell disease is caused by a mutation in the beta globin gene. And it's very common in people of West African ancestry. And the reason it's so common is that people who have one copy of the sickle cell mutation and one normal are protected from malaria, which is a really deadly disease in Africa. So even though having two copies of the sickle mutation can cause severe disease, it's maintained in the population. So here's a little bit of math. You can feel free to ignore this, but those of you who are interested, I'll just get into a little bit more detail here, which is one of the ways from the whole genome sequence data that you can find the signatures of local adaptation to different environments. Is we use something called an FST statistic. This is a way of just looking at differences in frequency of different variants, okay? So let's say we're gonna compare these three populations and we're gonna look at all the variants in the genome. And we wanna ask the question, which variants are very different frequency in the, say, Baka rainforest hunter-gatherer group compared to the other African groups? And the other African groups are kind of similar to each other at this region. And that way you're finding things that are really more specific to that population. You look at the tail of the distribution, so you look at the ones that are most different, and you ask the question, what are the genes that are nearby, and what are they doing? And that might give you a clue about local adaptation. And when we do this for the different groups, we see a lot of potentially interesting examples of local adaptation. I'm just going to highlight a few in some of the hunter-gatherer groups. So let's start with the rainforest hunter-gatherers in Cameroon. And we could see that there's selection near genes that play a role in bone growth, which is really interesting because they're known for their short-statured trait, which is thought to be an adaptation to their tropical environment. Let's now look at the Hadza hunter-gatherers from East Africa. Well, here we see something really different. We see that they're very different at variants near genes that play a role in heart muscle development. And you could imagine how this may be adaptive in these populations that are going very long distances. They walk very long distances in very arid conditions with little water. So this is something we're following up on. We're trying to find what these variants are. And that might also give us a clue both about normal heart function and also when things go wrong, what may cause types of heart disease. Okay, next I'm going to tell you a story about, or a study I should say, that was done by Mike McQuillan in my lab. And here we're looking at something called a SNP array, a genotyping array. So these, instead of sequencing the whole genome, we're looking specifically at regions of the genome that are known to be variable. So at millions of variants, and we genotype people from across Africa, about 1,071, and on the lower left, this is showing the distribution. And then we have this admixture plot that I told you about. So you can just see, again, there's a lot of variation here. Now I'm going to tell you a little bit about another method that we use for looking at signatures of natural selection. And this is a way to detect very recent positive selection. So let's say these different variants, these dots represent different genetic variants that are segregating in a population. And let's say that a mutation occurs that increases the fitness of the person who has it. What do I mean by that? I mean that they have more children and their children have more children and so on. Therefore, it's going to rapidly rise up in frequency in the population, as shown here. And what's going to happen is when that happens quickly, it drags with it the neighboring variation. And so at places where people are homozygous, they have two copies of this mutation that's adaptive, they are homozygous at these neighboring mutations as well. So we call this, one of the approaches is called extended haplotype homozygosity, is one of the methods that's used. And there's a particular test, a statistical test called the IHS test that can be used to detect that. Okay, so we apply these two different types of tests. This is based on allele frequency differences, and this one's based on the extended haplotype homozygosity. We take the top 1% and we say, what are the genes nearby? What do they do? Okay, and that's going to give us this clue. So I know this is very small. You may not be able to read this in detail, so I'll tell you what it is, but something really stood out to us because we looked at all these diverse Africans and we start seeing the same pathway called, it's genes involved in alcohol dehydrogenase activity. And it's coming out in all these different Ethiopian populations that speak Afroasiatic languages. And we don't typically see that. That's very unusual. So we wanted to follow up on this. So let me tell you a bit about this. The alcohol dehydrogenase gene family codes for enzymes that catalyze the metabolism of ethanol into the toxic intermediate acetaldehyde, shown here. There are non-synonymous mutations. So this is mutations that alter the protein. And this is one that's well known. It's called ADH1B48 His. And it increases the catalytic activity of the enzyme, leading to a buildup of acetaldehyde. And there have actually been genome-wide association studies that show that this mutation is protective against alcoholism, because actually you get pretty sick if you have this because you can't metabolize the alcohol. And for example, it's known that it's very common in East Asia, and it can cause flushing after people drink alcohol. Now, people had shown that this is a target of natural selection in East Asia, and it was hypothesized based on the age of the mutation that it correlates with the development of rice agriculture and an increase in fermented foods and beverages. That was hypothesized to be the selective force. Well, what about the Africans? Well, these are Ethiopians. They have a lot of fermented food and drinks as well. But I want to point out that this alcohol dehydrogenase pathway is really, or gene is important in a number of metabolic pathways. So it could be doing a number of different things, for example. All right, so here are those two statistics that I told you about that detect signatures of selection. And this red line represents anything above that is like the top, it looks like 0.5, 5 percentile. And you see this big region, okay? And it's overlapping, let's see if I can find the gene. The alcohol dehydrogenase gene is in here, but so are some other genes as well. So this whole region appears to be under selection, hundreds of thousands of nucleotides. Now, what's really interesting is this is what we see in populations that practice agriculture. Now, let's look at a population that speaks the same language. It has the same inferred genetic ancestry, but it's not agriculturalist, okay? So they're either hunter-gatherers or pastoralists. And we don't see that at all. This signature is specific to agriculturalist groups. That's very, very unusual. So here, I see I'm going to have to speed up a little bit. Here we see this variant is common in East Africa. This is that signature of extended haplotype homozygosity that I was talking about. And there are other genes nearby and other mutations that also might be targets of natural selection. So to look at the origin of this and how it relates to the East Asian mutation, we constructed what's called a haplotype network. So a haplotype, if you look along a little piece of a chromosome, how the different variants are arranged in that piece of the chromosome, say over like 40,000 nucleotides, we call that a haplotype. So each circle here is a haplotype. And this is the haplotype in which the East Asian mutation is on. And a very different and divergent haplotype is the one that the Ethiopian mutation is on. But you could see it's also common in not common, but it's present in Europeans and in some Middle Eastern populations. So we wanted to know if maybe did it arise independently in Africa, or was it introduced for migration? And one way we could do that is something called where we look at the local ancestry sliding along the chromosome. So this is that region where these alcohol dehydrogenase genes are. And if you've got purple ancestry, it's sub-Saharan Africa. If you've got yellow ancestry, it's non-African. And what we see is an enrichment for non-African ancestry right in that region. And in fact, we can estimate that the timing of admixture in Ethiopia of non-Africans with Africans, and we can estimate that this mutation we think was introduced around 2,000 years ago. So we think it came from outside of Africa to Ethiopia, and then it was under strong selection only in agriculturalist groups where the frequency is very different than in groups with the same genetic ancestry but different diets. So that's a really unusual pattern. And this supports a hypothesis that agriculture somehow shaped the evolutionary history, but we still don't know exactly the selective force. I'm going to spend, say, another 5 to 10, probably about 10 minutes on this next part, and then I'll stop. I want to talk about one other adaptive trait, which is skin color. So here in blue, this is showing the level of melanin. Melanin is the pigment in skin that causes more darkly pigmented skin. And we see that the levels correlate with UV levels. So it's thought that when our ancestors left Africa and went to higher latitude, there'd be selection for lighter skin. Possibly it's hypothesized to help facilitate production of vitamin D, which is synthesized in the skin in response to ultraviolet radiation. And in regions with a lot of UV, on the other hand, it would be protective to have more darkly pigmented skin. Okay, so how do we do this in the field? We actually use something called a spectrophotometer, this machine here. We shine it under the arm, which is a place where you don't get a lot of sunlight. And based on the wavelength of the reflection, you can infer the melanin levels. And we see a lot of variation in Africa. The most lightly pigmented population in Africa is the San. And this is not due to, say, admixture with non-Africans. Okay, this is naturally occurring light skin color. The most darkly pigmented people in the whole world actually originate from South Sudan, and they speak Nilotic or Nilo-Saharan languages. We then did this genome-wide association study, and this was published in Science, back in, I think, 2017. And this is a very small number of people. 1,570 is not a big sample size, but we still found 8 independent associations at genome-wide significance. I'm just going to quickly step through. So the first is a coding change in the protein. It causes a change in the protein at a gene called SLC24A5. The light-associated variant is at pretty much 100% frequency in Europeans. It's also common in Pakistan and parts of India, and then it's common in East Asia. And this actually was the very first mutation ever found to play a role in light skin color in Europeans. So this is like a classic known mutation. So we look at the haplotype network, and this time, unlike what I showed you before, the East African variant here is on exactly the same haplotype background. So meaning the last time I talked about the East Asian versus Africa. Here, this is the same haplotype background. So we think it was introduced from outside of Africa and then became more common, perhaps due to selection. All right, the second region was a gene that had no name at the time. We're the first people to characterize this gene. If you go to PubMed, we're actually the first ones in there. I'm very proud of that. And we found two independent associations, one in this region upstream that regulates the expression of MFSC12. The other is in this region. We still don't know exactly what it does. We suspect it's impacting gene regulation. Let's look at the variant up here that's associated with skin color. The light-associated variant is common in Europeans, throughout basically Eurasia, and in the East Asian or East Africans, and in the San, who are lightly pigmented. The dark-associated variant common in Sub-Saharan Africa, South Asia, Australia, Melanesia. So our collaborator at NIH, Bill Pavin, knocked out this gene using CRISPR technology in a mouse and had a dramatic impact on the pigmentation. And now this has been verified in a number of studies, and it turns out this gene even plays a role in risk for melanoma. So what's encouraging, though, is that even with relatively small sample sizes, because of all the variation in Africa, it can be really informative. And then together with Yun Song and Ethan Jewett, they looked at what's called the gene genealogy. Think about it like the phylogeny I showed you if you unwrap this. And again, at the ends here are genome sequences. If they have the light-associated variant, it's this open circle. These would be the dark-associated. They're color-coded by geographic region. And basically, it's a way, we're looking at the gene genealogy. It's a way to estimate the age of the different variants. So in this case, the ancestral variant is the one associated with light skin color, but both of them are very old. So the derived mutation is still almost 1 million years. All right, now we go to DDB1. These are two independent associations near this gene. This gene plays a very important role in DNA repair caused by damage by the sun. And in fact, children, let's say who are born with mutation in this gene, have something called xeroderma pigmentosum. They can't be exposed to sunlight because they can't repair the DNA damage. They get skin cancer. What does this have to do with pigmentation in humans? I don't know, but it altered the color of tomatoes, so we figured it has something to do. I'm not going to have time today to tell you about a more recent study we did that was published in Nature Genetics. We'll save that for another time. But in that study, we actually used some more modern techniques to look at interactions between regulatory regions and the target genes. And what we found is that while DDB1 is important, the most critical genes are these two right here. And in fact, CYB561A3, that turns out to be probably the most important. Okay, so here we use another test to look for signatures of selection. This is based on the allele frequency distribution, meaning this is the number of singletons, doubletons, tripletons, and so on. The blue line is what you expect under a neutral model. And if there's positive selection, you expect an excess of rare variants. If there's balancing selection, you expect an excess of common variants. And what we see here, these are Europeans, Chinese, this is West African, these are Nilotic, these are San. This really massive signature of positive selection, this negative to G minus D value, just in Europeans and East Asians, overlapping hundreds of thousands of nucleotides. And in fact, if you look at the heterozygosity, that's a measure of the genomic diversity, it is wiped out in Asia and almost wiped out in Europe. And if you look at the haplotype variation, this is over 200,000 nucleotides. There's this one long common variant outside of Africa. And if you look at the gene genealogy, you see that all these lineages coalesce around 60,000 years ago, right around the time of migration of modern humans out of Africa. So I think this is a rare example where there's something on this chromosomal region. We don't know what it is, but it's really important. And it rapidly swept to almost 100% frequency at some point in non-Africans. And then the last region is a gene called OCA2 and HERC2. This is known, OCA2 is well known to play a role in skin color. Mutations cause albinism. 2 of these variants were in upstream regions that regulate the expression of OCA2. They arose independently from some known variants in Europeans. But this other variant was what we call, it wasn't impacting the coding region of the gene, and it wasn't impacting gene expression. So what was it doing? It was causing alternative splicing. So what it does is it causes the light-associated variant is associated with a shorter gene transcript, and it actually knocks out this exon 10, one of the exons. It knocks out this third transmembrane domain of this protein, but it still codes for a protein, but it has a dramatic impact, you can imagine, on the function of that. And then the last thing is that the only place where we saw these dark associated variants are outside of sub-Saharan Africa or South Asia and Australia, Melanesia, where we see people with darkly pigmented skin. And we were able to show that at least in this case, these variants actually originated in Africa, and they were brought out during the out of Africa migration. So in summary, half the cases, the ancestral allele is the light associated allele. And if we look at the age of the derived, the newer mutation, it predates the origin of modern humans in most cases. So there have been light and dark variants, associated variants evolving in Africa for a long time. And I'm going to actually just end there. I'm going to jump to my thank you acknowledgements slide here at the end. And just say thank you to the many people from my lab and collaborators, including our African collaborators who have contributed to the study. And I am happy to answer any questions that you may have if we have time. Thank you.
Great. Thank you so much, Dr. Sarah Tishko. That was a fascinating talk. So we do have questions from the audience. So I'll start by this question from Marcela Elizalde. So it's in Spanish, but I'll translate for you.
Okay.
So greetings from Eastern Mexico. In your research, have you found possible changes in human evolution due to viruses? I'm asking because of the recent SARS-CoV-2 pandemic we experienced worldwide.
Yeah, that's a great question. Do we see like evidence of, say, natural selection impacting regions of the genome that may play a role in viral susceptibility or resistance? And I don't have any great examples from my lab, but we know that these exist. So certainly other people have shown that absolutely there is nothing that is a stronger force of natural selection than infectious disease. Number one, because that's the one thing that can cause deaths of hundreds, thousands of people. It could wipe out an entire population. So if somebody has natural resistance, it's going to rise to high frequency. And just anecdotally, like people have observed that in the rainforest hunter-gatherers, that they're not as susceptible to some viral infection. And we do suspect that there might be something about their genomes that's different. We're actually studying that now by doing something called single cell sequencing of the white blood cells before and after immune challenges. So we're looking into this right now. And actually, though, speaking of SARS, you know, there is this example that I think it was Fonte Pebo's group, right, about the intergress, something, a region that was intergress was introduced by the interbreeding with Neanderthals, if I'm remembering correctly, that plays a role in susceptibility to SARS, you know, CoV-2. So, yeah, absolutely, those exist.
Great, thank you so much. So another question we have is from Sandrine Mirales, Universidad del Mar de Huatulco, Oaxaca. It was recently suggested that a population which diverged before the split of Neanderthals later contributed 20% of variants of modern humans. Why would their contribution be higher than that from Neanderthals despite their great divergence time and likely lower hybrid fitness?
I can't answer because unfortunately I'm not familiar with that. I don't know if somebody else is familiar with that study, but I'm not. So unfortunately I can't address that.
Sure. Okay, so we have another question from Jacqueline Nava. It's what is causing the population crashes for hunter-gatherer communities? And again, take the question as you see fit. If it's also misunderstood, you can address.
No, that's a really good question. And what really interests me is, for example, if you compare the Huds and the Sandawe, so they both speak with the clicks, they're indigenous to Tanzania. The Sandawe for whatever reasons, when the people, the agriculturalists and the pastoralists migrate to that region, they chose, or for whatever reason, maybe they didn't have a choice, you never know, but they married or admixed or with these other groups. Okay, so that now when you look at their genome, it's like a mixture of all these different populations together. And then they adopted some of the agriculturalist and pastoralist technologies, but they still hunt a bit. So they still have that culture of hunting, but their population size grew. They're now at like probably about 100,000, something like that. But the Hadza, perhaps for cultural reasons, decide, no, we're not going to assimilate. And so they have intermarried a bit, but they decided, no, we're not going to adopt these ways of the other people, and decided to keep their traditional lifestyle for as long as they can, because they're being encroached upon now that a lot of their land has been taken away. So there's very limited animals available to them right now. So we're hoping for the best for them.
Muchas gracias. So next we have a question from Javier Ramirez, which is, it's generally about informed consent, so maybe you can comment more on it. The question in specific is it really possible to achieve informed consent in isolated communities? If you could just comment more on how you go about that.
Sure. I mean, I would like to think so. We can't be naive that, these are people who generally are not going to have a high level of education, but that doesn't mean that if you start talking to people about, for example, family patterns of relatedness, or people in your family look more similar than between your family and another family, or between this group and that group, or between me and you, and so on. People get this idea. And interestingly, in Africa, at least, keep in mind, can be very culturally specific, because in Africa, when they have the sense of their ancestors are in their blood. So if you say there's something called DNA and it's passed on through the blood and it reflects your ancestors and your history, they're like, yeah, we totally get that. Again, it's how you explain it. You have to explain it in basic ways that hopefully people will understand. I think they do. I guess the one bit of encouragement I have is that group, the Sandawe I mentioned. They're in a pretty remote area. I went back there. I try to go back and return results. So we go back the next year, and this guy comes up to me and he has a pamphlet that's given out by missionaries, like Christian missionaries. So it's not going to be about evolution, but it happened to be about science. And it had a double helix in there. And he pointed at that. He said, so that's what you're studying, right? And he goes, you could figure out where, Black people from the Americas came from in Africa. And I was like, yeah, like, I think he gets it. So we do our best.
Okay, thank you so much. So next up, we have a question from Alfredo Munoz Garcia. And the question is, it's a general question. I think we can get some of your comment on it, just relating to the talk in general. So Dr. TISHKOFF, considering your research on genetic diversity in African populations and its role in understanding human evolution in general, given your extensive experience in this field, how do you assess the significant importance of the interaction between natural selection and demographic factors in the emergence and distribution of complex adaptive traits in today's interconnected and globalized world?
It's all important. Absolutely. One of the challenges we have as evolutionary geneticists, right, is distinguishing. When you look for these signatures of natural selection, sometimes they can be caused by demographic history, right? So it can be very tricky to figure that out, but there's no doubt that both are, because you can have some new variant that's introduced by migration, mutation. Sometimes there's random drift, just random fluctuations. So the fact is both are contributing, and I believe that both are likely contributing to common disease, but it depends on the disease. That's very controversial, because typically common disease is caused by hundreds, if not thousands, of variants. And so there may not be any that are under really strong selection. Skin color is a little unique because there are variants of really strong impact. And so you can pick those up with scans of natural selection. It can be a little bit more tricky, and there are certain tests that are used to identify what we call polygenic selection on many genes that contribute to complex traits. But I believe it's a balance that's happening, but it's very tricky to distinguish the role of demography in selection.
Okay, thank you. So another question, Sir Janet, I think maybe you can talk a little bit about it. It's from Manuel Garcia. So extremely hot days like the ones we're currently experiencing lead us to believe that prolonged periods of heat or cold could impact habitat development. Well, the question in particular is, do you think we're close to experiencing these impacts on the planet due to climate change? I know your talk wasn't about that,
Yeah.
but in Africa, like, is there anything
Mhm.
related to that you've been looking at, like climate's effect on adaptation?
Yeah, so I think the answer is yes. We are going to be impacted by climate change, and the places that will be impacted the worst probably are going to be some places like in Africa and elsewhere where it's going to have extreme weather impacts, and that's also going to impact things like infectious disease and a number of health-related issues. So it's something that we should be really concerned about. So while I'm not studying the recent impacts of climate change, we are interested in how some groups in Africa, for example, have adapted to extreme aridity, like very hot climate. So some of the pastoralist groups we study, and the Hadza, for example, and then the San, they're in deserts. Like they don't even have access to water often. They're getting it from roots. How did they adapt to those environments? That's something we're really interested in because it'll tell us something about how has, nature resulted in these adaptations to a changing climate, and maybe it'll give us a clue about what we can expect in the future.
Thank you. So another question we have is from Angel Luna, Iztacalaca, UNAM. You showed evidence for natural selection on skin color variants. Now that humans migrate so much and mix with each other, is there any evidence of selection on allele frequencies in this modern context? If you can just extrapolate a bit there, what you already talked about.
So no, first of all, we're on such short time scales. We're talking the last, few hundred years, right? So there's not a lot of time for selection to act, although some people are looking. There's some argument that in like recently admixed populations in the Americas, for example, could there have been selection even very recently? But we don't have any evidence, certainly for skin color. And again, there's cultural adaptations. People now can wear long clothing. They can wear, have skin protection, right? Sun protection on their skin. So I don't think there's as strong, but it depends where you are because again, the populations that I study in Africa, and actually one of the groups that we're now studying are people with albinism, which is remarkably common just in certain ethnic groups in Africa. And I'm really, really curious about why that is, because in theory, it shouldn't be that common. And when I've met with the albino populations in East Africa, for example, they can't afford sunscreen. They sometimes can't afford sunglasses or long, sleeve clothing. There is selection going on, no doubt, in many regions particularly where there's poverty and they can't afford things like sunscreen. So I think it does still occur in some populations, but in urban populations, probably not so much.
Thank you. So we have another question from Javier Ramirez, which is, how does your research reduce inequality and access to genetic knowledge, and what impact could it have on clinical developments? So if you talk a bit more on if you've seen some more examples into that.
So I think if we don't study these diverse populations, they can't benefit from any knowledge that may be gained, right? I mean, if you're not in the game, so to speak, if you're not even included, we don't know what we may be missing. I think the knowledge that we get is not just relevant for those particular populations, but it can be relevant for all of us because one classic example is a gene. I hope I'm getting this right. PCSK9 or PKSC9. I'm suddenly getting it mixed up in my head. But this was a gene that was found in families of African ancestry, and it was associated with LDL, like very low LDL levels. And then this became the target of a drug that Regeneron made, a therapeutic that's extremely effective in everybody, right? It doesn't matter your background. But for things like the genetic risk scores, you have to study these diverse populations. That's the only way that you're going to have more effective risk estimates. And if you have more effective risk estimates, it means, for example, let's say someone's at risk for heart disease, very high risk. You could say, okay, you better start on this treatment very early, or you got to be really careful about exercise or whatever. So both it could lead to better diagnostics and possibly better therapeutics. So I think it's really important for those reasons.
Great. Thank you so much, Sarah. So I think, do we have time for a couple more questions or? We have to close.
It's up to you guys.
Okay. If you're available, there are a couple more questions. So
Okay.
if you, okay. So another just sort of more general question, which would be good to have your comment on from Alfredo Muniz is, will we tend towards homogenization of traits or greater evolutionary diversity?
That's a good one. I don't know, because on the one hand, you could think of like there's so much, globalization, right? And people are intermarrying and things like that. On the other hand, we know that that could actually introduce more variation, right? So I'm afraid I don't have a great answer for that one. That's like, I think the philosophers might have to think about it. I don't know. That's hard. I can't predict for sure.
Thank you, Sarah. So I had a few questions just generally from your talk. Was if you can just comment for the audience on what can cause some African groups to have more diversity than other groups, as you showed, what can cause some pathogenic variants to exist in only some groups in Africa or in the world, not anywhere else in the world. So
Yeah.
from that.
So the levels of diversity are going to be reflecting a couple of things. So one is the fact that these populations have been around for a long time, right? So there's been time to accumulate variation. And then the amount of variation that's maintained in a population also reflects things like the population size. So sometimes if they've been bigger for a longer time, that also can result in more general variation in the population. In terms of population specific, it can be due to random mutations that occur after populations have diverged from each other. They've migrated to different regions, random mutations may occur. And then we can also have adaptation to these different diets or environments, climates, pathogens. So again, if there happens to be, let's say, a pathogen that's specific to a particular region and there by chance are mutations that provide some resistance, it's going to be common only in that population or that region. So that's how it would happen.
Great, thank you so much. And just the last question from me would be, with the caught my attention, one of the things you mentioned was that if I caught it right, the rainforest hunter-gatherers, there's adaptation for short stature. So just could you talk a bit more, why would short stature be adapted for in the tropical environment?
So we don't know. There's lots of speculation. This is a trait that we see in different regions. You see it in the Philippines, you see it in South America, different places that are in people living in the tropics. You'll see this short stature trait that is evolving in these different regions. So all kinds of theories, but one of them has been that it has to do with the very short lifespan in these populations. And it is true that when we go there and we just ask them demographic questions, they are dying at a really young age. So demographers have shown that the expected, there's like a 40% chance of living to age 15. And then if you make it to age 15, average lifespan, it's something like 25. I mean, it's like really, really low because of a lot of infectious disease, a very harsh lifestyle. They climb up very tall trees. Sometimes they fall out. It's just a very harsh lifestyle. So the idea is that if you're dying at a very young age, there'd be selection to reproduce at a younger age. So possibly that puberty could be reached earlier and there might be a lack of an adolescent growth spurt. But this has been disputed. Some people have said, no, that's not true. I think we don't know. We actually have been studying traits associated with puberty in that population. So we hope to be able to address that. That's something we're still looking at. So we still don't know.
Great. Okay. So unless there's any questions from Araxi or Jaime, I think that's all we have from the audience.
Okay, well, thank you so much. I've really enjoyed being here. Wish I could be there in person, but maybe another time.
Thank you so much, Dr. TISHKOFF. It was a great talk and lots of engagement from the audience. Muchas gracias a todos y todas por asistir. And nos vemos la siguiente semana, la próxima charla.
Bye-bye.
Bye.